Okay, so you just watched this Bioflix video, maybe hopefully a couple of times, taking notes, making sure you understand what it's talking about. So let's get into it a little bit more in depth. So eukaryotic cell division is going to be divided into two general parts. One is mitosis, which is when you are pretty much breaking down the nucleus, copying everything, and putting it back into two new nuclei. And then cytokinesis, which is when you divide the cytoplasm. So you actually pinch it out, make two different cells. Gametes are going to be produced by a different type of cell division called meiosis. So this chapter, the rest of it, where we're talking about mitosis, it is not happening in gametes. This is the rest of your somatic cells in your body. Meiosis will give you non-identical daughter cells that have half as many chromosomes as the parent cell, okay? So we are now going to focus on mitosis and not meiosis. The cell cycle is going to be interphase. Interphase is when the cell is growing, and interphase, the cell is copying its chromosomes, and interphase is when the cell is preparing for cell division. So interphase is G1, or gap, or growth 1, so the cell is just growing. It is doing whatever its job is. So if it's a muscle cell, it's helping your cells move. If it's a skin cell, it's functioning to protect your cell. S is synthesis, for DNA synthesis. So we're still in interphase. In the S phase of interphase, we are copying DNA. And then G2, when the cell is still growing and acting, doing whatever it's supposed to be doing as a cell. Then you get to the mitotic phase, and in the mitotic phase, you have mitosis, when the DNA is dividing, and cytokinesis, when you have one cell actually split apart into two genetically identical daughter cells. So at the end of mitosis, you have one cell that is split into two cells, and those cells have identical DNA. They have the same function. They're going to be doing the same thing. You wouldn't be able to tell which cell was a parent and which one was a daughter because they are identical to each other. Okay, interphase is about 90% of the cell cycle, so most of the time your cell is going to be in interphase, and then that is G1, S, and G2, or the first gap, synthesis, and the second gap. The cell is growing during all three of these, but chromosomes are only going to be copied in the S phase. So you start with one copy of chromosomes, one copy of each chromosome in G1, then by S you have sister chromatids. In S you're making, then in G2 you have sister chromatids, and in mitosis you are splitting apart those sister chromatids, and by the end of cytokinesis you have two different cells that each have a copy of every single chromosome. Mitosis is broken down into 5 stages, so I think of PP mats, so like trying to train a dog, house train a dog or something, a PP mat. So P for prophase, P for prophase, P for prometaphase, then metaphase, anaphase, telophase. So you need to be able to tell me those in order, and then you also need to be able to tell me what is going on in each step. So here we have a cell in G2 of interphase. It's got a lot of DNA. Its DNA has been copied already, but they are not condensed, so they're all just kind of spread out, relaxing. You still have a nucleolus because in G2 that cell is still doing stuff, so it's still making ribosomal DNA, so you've still got a nucleolus. You've still got a nuclear envelope, so you still have a very distinct nucleus. You have a nice plasma membrane, and centrosomes are going to be arranged with centriole pairs. Okay, so that is interphase by the end of G2 when it's ready to start mitosis. By this point, that cell that hits G2 is committed to go all the way through mitosis. It can't just hang out anymore. Then in prophase, you're going to start seeing early mitotic spindles. Your chromatids are condensing. You can see sister chromatids, but your sister chromatids are still connected, and there is still a nuclear envelope, so that DNA is still within the nucleus. Then in prometaphase, your nuclear envelope is fragmenting away. The kinetochores on your DNA are connecting to kinetochore microtubules, which are connected to the centrosomes, and those centrosomes have moved to opposite sides of the cell. Then in metaphase, you have a metaphase plate. The metaphase plate is when those chromosomes are lined up on this imaginary plane. So you can just pretend like you have a plate in the middle of it. The centrosomes are at spindle poles at the opposite ends. And all of your chromosomes should be attached at their kinetochores to the spindles. In anaphase, you yank those apart. Anaphase is a pretty quick thing because it's just grab those sister chromatids, and the sister chromatids are no longer connected to each other. So they are now individual chromosomes moving towards those opposite ends of the cell. And then in telophase, you have you're starting to create new nuclear envelopes around your DNA. You're starting to get a new nucleolus where you can start making RNA, ribosomal RNA. And you have where you can see this two cells are splitting, starting to be pinched into two. So this is an animal cell. We have a cleavage furrow. It's just like if you had a shoelace tied around the cell and you're tying it around to pinch it off. So that is a process of cytokinesis happening. So in this cell, telophase and cytokinesis are happening at the same time. And that's okay. That's a normal thing to see. Okay, so let's watch this bioflix.
This soccer player's knee
Oh,
will be as
we're
good as
not.
new in a few weeks.
Never mind. We watched that one, so we're not watching that again. Let's watch this actual micrograph of a cell undergoing mitosis. So here's a cell, so we're going to see it go through each of these phases. So it's growing. Now you can see its chromosomes have condensed. You can see very distinct chromosomes on here. So each of these are chromosomes. Okay, you can see they're lining up, getting a metaphase, forming that metaphase plate. See where they're just lining up. And then there's anaphase, yank those chromosomes apart. Oh, that's so pretty. Let's go back and watch it again. Yank, just anaphase. And then here is telophase, where you're getting cytokinesis and new nuclei forming. And now those chromosomes are not condensed anymore. They're loosened up again. So you can't see those distinct chromosomes. Okay, let's watch it again and see if you can tell for yourself when each of those stages are happening. Oh, this is beautiful, isn't it? Okay. I love micrographs. You look at anything under a microscope, it's going to be beautiful, isn't it? Okay. So the mitotic spindle, let's talk about that. We talked about it here, where we talked about these are our spindles. So the mitotic spindle, this is a structure made of microtubules. You remember talking about microtubules when we talked about the cytoskeleton. And the mitotic spindle controls the movement of chromosomes. So when chromosomes are lining up on that metaphase plate, it is because mitotic spindles are putting it there. When they are being yanked apart during anaphase, it is because mitotic spindles are moving it there. So all of the chromosome movement that we see during mitosis is controlled by the mitotic spindle. In animal cells, those spindle microtubules get assembled beginning at the centrosome. And the centrosome is kind of just the microtubule organizing center. So that is the, Kmart manager that is very important and says centrosomes are going to be made here and can kind of bosses that whole thing around. The centrosome will replicate during interphase. So you have centrosomes available because your cell has just gone through interphase. And during interphase, you'll form two centrosomes that will migrate to opposite edges of the cell. So let's go back and look at this. So you see these centrosomes getting replicated during interphase, and they're moving to opposite ends of the cell. Start moving during prophase, and by prometaphase is when they're actually fully moved over. Okay. The aster is a radial array of short microtubules that you have at each centrosome. And so the spindle will include the centrosomes, the spindle microtubules, and the asters. So if we go back to this picture, I should just put it in. Here it says, where did it talk about asters? Oh, I thought we talked about asters on this. Oh, here, this is the aster in this part. Okay, so looking at each of these steps a little bit more. During prometaphase, some of the spindle microtubules will attach to kinetochores of chromosomes. So here are chromosomes, and you can see these attaching to the kinetochores. And the kinetochores are proteins in the middle of the centromeres, the proteins that are attached to the centromeres. And at metaphase, the chromosomes line up at this imaginary plate called the metaphase plate. And the metaphase plate is just right in the middle, halfway between the two different poles of the spindle. In anaphase, the cohesins will be cleaved by an enzyme called separase. And so getting tongue twisted. Separase will separate those, by separating the cohesins, it pulls apart those chromosomes. So in anaphase, sister chromatids will separate and move along the kinetochore, those microtubules, to opposite ends of the cell. And the microtubules will shorten by depolymerizing at their kinetochore ends. So these microtubules actually start depolymerizing, they start breaking down, pull these chromosomes to the opposite centrosomes. And then in animal cells, cytokinesis occurs by a process known as cleavage. So cleavage is when you just have this ring of microfilaments around the cell that just pinches in, just like taking a shoelace and tying it apart. So you get this thing called a cleavage furrow, or this is a cleavage furrow here. And that is just going to pull those two cells apart until you have two daughter cells. In plant cells, because you have to form a new cell wall between those cells, it works a little bit differently. So in plant cells, you get what's called a cell plate. So a cell plate is where you are gradually forming cell membrane and cell wall between the two sides of the cell. And so that cell plate is going to be something that tells you you're looking at a plant cell, and a cleavage furrow is going to be something that tells you you're looking at an animal cell. So two different ways of doing cytokinesis, depending on if you are an animal cell or a plant cell. So let's look at cytokinesis a little more.
In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating two identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the two daughter cells. The cell plate's contents join the parental cell wall. The result is two daughter cells, each bounded by its own continuous plasma membrane and cell wall.
Okay, so while the steps of mitosis itself are pretty similar between plant and animal cells, cytokinesis is very different between those two. So you need to be able to describe to me what a cell plate is. You need to be able to describe to me what a cleavage furrow is and what types of cells you would see either one in. Okay, and then what happens to those two daughter cells produced after you finish the cell cycle? That is going to depend on what type of cells they are. Some cells are constantly dividing, things like your red blood cells, things like your skin cells. And those cells that are constantly dividing, once they finish the cell cycle, they might immediately just go right back into another round of cell division. In an embryo, in an early embryo, many types of cell types are dividing quickly. so that that embryo can go from one cell to, you know, all the cells you need in a person. Many cells in a tumor are dividing rapidly, and that doesn't mean the cell division goes faster, it just means that they are constantly dividing. So they finish a cell cycle and it starts again, and soon that one cell turns into 2, turns into 4, turns into 8, turns into a large tumor pretty quickly. Other types of cells divide very slowly, or they don't divide at all. So your brain cells, your nervous system doesn't divide much. Muscle cells don't divide much. They can exit the cell cycle and go into what's called GO phase. And in that GO or G0 phase, a cell is not preparing to divide. It is just doing its job. So your brain cells are, you know, acting as a brain. They're not dividing themselves. Your muscle cells are acting as a muscle. They're not actively preparing to divide themselves. So the three main functions of mitosis, all of what we just talked about, one is for growth and development. So to be able to just grow from being a small child to an adult. One is tissue renewal. So that video where we saw a soccer player that scraped his skin. His skin cells will go through mitosis to renew that wound, to heal it. And then reproducing organisms to carry on the species. So some, you know, protists go through mitosis as reproduction. Okay, this is a lot of words, but this is an important topic, and that is programmed cell death. Programmed cell death is not a cell dying because it's infected or something wrong with it. Programmed cell death is when a cell dies because that is how that organism develops. So in programmed cell death is a carefully controlled, it's programmed, right? It's carefully controlled way. So we're talking about how our fingers form. If you've ever seen little pictures of, you know, development, it starts as kind of like a webbed thing. And that webbing, instead of staying there, those cells that make the webbing have to undergo programmed cell death to give us individual fingers. And so the cells between your developing fingers were instructed to die while you were an embryo. Otherwise, you'd still have webbed hands or maybe just paddles of tissue with no fingers. And so the cells between your embryonic fingers, those cells, that programmed cell death is called apoptosis. So that PT, the P is silent like pterodactyl. So apoptosis. Apoptosis is a common form of programmed cell death where cells undergo a specific self-destruct sequence when they receive certain cues. So certain signals tell those cells between those webbed fingers that they need to die and let your fingers turn into fingers. And it's very neat and clean as opposed to, you know, like an infected cell that might go crazy in its death. Programmed cell death is very carefully controlled. And it involves the death of this individual cell, but benefits the organism as a whole. So apoptosis will let your fingers develop. We can take advantage of apoptosis to eliminate potential cancer cells. If your immune system is able to identify a potential cancer cell, that cell can undergo apoptosis so you don't develop cancer, or even medically if we are able to take advantage of apoptosis to kill a suspected tumor. Okay, the opposite of programmed cell death is necrosis. Necrosis is when cells are damaged, so injury or exposure to some toxin, and when they just like explode, they spill their guts, blood, and die everywhere. And that's a lot because this plasma membrane the plasma membrane just loses control. So ions, waters, it leaks, it swells, and it often can also cause inflammation. Inflammation is not always a pleasant experience. So you don't really want inflammation, an inflammatory process going on in your cells, in your tissues. So cells that undergo apoptosis will go through a different and more orderly process. They shrink. They develop little blebs, which are these little bubbly things around it. Their nucleus, their DNA gets chopped up, and it just very neatly fragments into these little packages. And then those chunks will be eaten by your phagocytic cells. If you remember, phagocytic cells are your white blood cells, like your macrophages, that can eat that debris and just break down those little blobs so that you no longer have those cells that just went, you don't have any remnants of that apoptotic cell. Okay, many cells in the human body have a built-in ability to undergo apoptosis, the same way we have a built-in ability to copy DNA or break down sugar. And three important ways that we really care about apoptosis happening. One is by some cells need to be deleted during development. So like when we were talking about that webbing between your fingers, those webbing needs to be deleted. Some cells are abnormal and can hurt the rest of the organism. This should be cells that are infected with the virus, cells that have DNA damage that could, possibly turn into cancer or some other harmful, do something that harms your body. And then cells in an adult organism may need to be eliminated to maintain balance. So we are constantly making new platelets and breaking down the old platelets. So that makes, by getting rid of the old ones, you are going to make way for new cells, and you're also going to get rid of cells that are only temporarily needed. Okay. In many organisms, programmed cell death is a normal part of development. So here is a growing mouse paw. You can see all these webbing in between it, and this here at each star is where each cells are going through apoptosis. We are not going to read this stuff. I just think this picture is beautiful. So just understanding apoptosis is a normal part of development. Okay, I think we are going to finish these last 10 or 11 slides in part 3 because I don't really want this video going much longer than this. So I will see you back for part 3 in a little bit.